Okay, introduction to routine urinalysis. So, a little bit of history. We used to have these pissy prophets in the 16th century, and they claimed to be able to see all these diseases in urine. Like they could look at a urine and tell you what disease based on, the odor, the appearance, the taste of the urine. They really weren't too far from the truth at times, but that's 16th century. It was kind of like made up more. So they used to, before when we got married, they used to have to examine the urine. I'm sure it was a big patriarchal thing, like, nope, you're not getting married, you don't deserve it, you're positive for whatever, they made things up. But it used to be important that you have to get tested before you could get married. One of the earliest actual known records of a urine test, however, so we kind of knew this all before, but they would actually take urine and they'd spill it on the ground and they'd see if insects would come. And if the insects were attracted to it, it indicated the presence of sugar. What disease do you think that is?
Diabetes.
Diabetes, right. So it was really the first recorded lab test we had was urine, spilling it on the ground and watching for bugs. I can think of a whole lot of errors for that, right? 300 little errors on that, but it was really kind of cool if you think back to it. We were able to know that. People knew it meant something, they just didn't quite know what it meant. So in the 20th century, we got Benedict solution, and this was the first time we could regularly, with consistent results, test urine for the presence of glucose. Okay, what we use currently to this day actually is kind of still, it's a copper reduction test, and it's still a similar method. But this was the first, in the 20th century, we first could actually have a credible laboratory test that was repeatable, and we understood the science behind it more. So today, we get these great strips. This is a multi-sticks chem strips, both kind of strips we have in the lab. They're very simple, they're inexpensive, yet they're pretty sophisticated because you can tell a lot of information about a simple dipstick. If you think back to basic lab skills, in basic lab skills, you guys all did dipsticks, right? Some of you guys might have just done ketones and glucose, some might have done a whole dip. It kind of depends on where you took the class, what strips we had available. We use this still today. A lot of labs, even their big instruments, are still based on strip technology. It's easy, it's a wave test, meaning anybody can do it. So we have our lab assistants and our phlebotomists and test specimen processors that are able to actually do these tests. What does it mean to be waved? What's a wave test?
It's very hard to mess up.
Very hard to mess up. Yep, it has to be very simple. Remember the criteria? Anybody's allowed to do it. You don't have to be a certified laboratorian, but it has to be easy to mess, or not, it has to be hard to mess up, not easy, hard to mess up. And the results, if you do mess it up, cannot be life or death. You have to, it has to be some, it can't have dire consequences if it does get messed up. Okay? So now with these screenings, these strips are great for screening. We can look for things like liver disease, urinary tract infections, diabetes, kidney disease, other infections. There's all sorts of different things we can tell liver issues by reading these strips. So there are 3 parts of a routine urinalysis. The first one is going to be your physical examination. We're going to look at color, clarity, odor, etc. We'll talk more about these on the next slides. I'll get into more depth. The second part is going to be your chemical analysis. That's when you're going to do the dipstick. We're going to look for sugars, proteins, bilirubin, cells, etc. Now, if there are certain criteria on the chemical analysis, it'll reflex to a microscopic. We do not do microscopics necessarily on everyone, but the microscopic is the third part. Now, microscopic, we're looking for the elements, the cellular elements, red cells, white cells, casts, crystals, bacteria, things that are solid, is what we're going to be looking for on the microscopes. So these are the 3 parts of a full urinalysis procedure: physical, chemical, microscopic. So, physical exam of urine, we're going to start with color. Straw, yellow, dark yellow, amber, those are all kind of our normal. Yellows, any gamut of yellows, all the way from straw to dark yellow, are considered normal. And I'm sure that we all know at different times in our lives that we've had straw urine and super dark urine, possibly even in the same 24 hours, right? You wake up in the morning, it's going to be darker because you're a little dehydrated. By the end of the day, if you're like me and you sip on water all day because you're talking all day and your voice gets icky, by the end of the day, it's pretty clear, pretty colorless. Yellow, the yellow urine is due to the pigment urochrome. Urochrome is created in the kidneys and it makes that yellow color. There's a couple other things that can affect the color, but the primary urinary pigment is urochrome. You'll see that again. Oftentimes, whether it's light or dark, it's determined because of the concentration of the urine, right? So we know that you get dehydrated, it gets darker. You're well hydrated, it gets lighter. You run one of those iron people races, and it gets pretty dark. You get pretty dehydrated, right? So, you know, and that's all normal. That's our body's way. That's the kidney's way of stabilizing everything. So it's all perfectly normal. The most common abnormal color we see is pink or red. Why?
Blood.
Blood. Blood is not normal in our urine. So we shouldn't see blood really. So if we do, that's our most common abnormal color. So urine clarity. We're looking at clarity. Clear, hazy, cloudy, and turbid. Those are the four that we use. These are all in the manual too. When you're reading through the lab manual, you'll see these. Clear, I can see right through it, read newspapers through it. Hazy, I can still see the print, but it's a little cloudy. Then it gets cloudy, then it gets turbid, you can't see through it. One thing we always have to remember is when we're dealing with a urine cup, sometimes the urine cups have a bit of turbidity to them. So you have to, in your brain, tare, you know, T-A-R-E. Remember when we did the scale, so you talk about tearing the scale? You have to tear that away. So how, is it clear other than the color of the container? So you have to mentally take away the color of the container to see how clear we're looking. Specific gravity. We have a whole lecture coming up on specific gravity, but it's the ratio of the mass of the solution versus the mass of an equal amount of water. So what's in it, right? Urine is just a little bit higher specific gravity than water. The specific gravity tells us whether or not the kidneys are capable of concentrating and how well they're capable of concentrating. Specific gravity can be anywhere from 1.003 to 1.035. It's a big range. Like I said, first thing in the morning, it's going to be higher. By late afternoon, if you're like me, it's going to be a lot lower. That whole range is normal. We'll talk about in that lecture why we worry about it and why do we study if the range is that large, right? We'll talk about it later. So that was physical. Second part is going to be your chemical analysis. We use these great things called dipsticks. They're called dipsticks because they're sticks that we dip into urine, right? Nothing fancy there. They're chemical reagent strips. So each of these pads is a different chemical reaction. They activate when you touch them against the urine, and the chemical reaction occurs, and the color changes depending on what you're looking at to a certain range. So they're plastic strips. They have one or more chemical test pads. As I said before, you guys did the ketones and glucose. Some of you just had strips with 2. Sometimes they're the whole 10s. Depends on what we're looking at. Each pad has different reagents that are going to react differently based on what it's looking for. As the chemical reaction occurs, they're timed. And when you get to a certain point of time, you read it. Now, I can tell you right now, I can dip a urine in it. 2 minutes, we read the leukocyte esterase. It turns purple if it's there. If I look at it 2 minutes, it's negative, and I put it down, but I look 4 or 5 minutes later, and it's purple, exposure to air made it purple, not the leukocytes. You want to read them at the time they're told to be read, or the color will continue to change just based on exposure and time. So it's really important to read them at a certain amount of time. When you read them at that part of time, it gives you a semi-quantitative result. So the pad might be 50, 100, 180, whatever. So if it's, for example, you see it's 100, it's between 100 and 180. That's the lower limit of that pad. It's between that and the next pad. That's semi-quantitative. Some of them are just positive, negative. That's qualitative. There are no exacts with urine dipsticks. We have some qualitative and some semi-quantitative. We don't have specific quantitative. So the different things that the pads do, we look at pH, blood, leukocyte esterase, nitrites, proteins, glucose, ketones, bilirubin, and urobilinogen. Each of these are going to tell us something different. We are going to go over each of these in extreme detail. You're going to learn everything you wanted to know, plus for these going future. So you don't have to memorize them now, but just know that each test pad tests for something different. So the procedure. When you're dipping these, you're going to take the stick and you're going to dip it all the way in the urine, so they're all underneath. I give it one quick swirl, just in case there's any air pockets, and then you pull it back out. You do not sit and spin it. You don't want that exposure because that'll just pull the chemicals off the pad. Dip, spin, pull it out. When you pull it out, you're going to pull it along the side of the top, and then we're going to blot it. You have these little orange biosquares on your desk, and you're going to blot it. The reason we do that is I do this before. This is the side of the strip, so there's 10 different strips on it. Well, the urine gets between them, and it kind of goes over the tops. And that'll actually be prolonged exposure if you don't. We want to blot that all out of there because otherwise it creates a bridge between the two pads. So you could have crossover on the pads, and just that extra exposure of the urine is going to cause the strips to not be as accurate. So we kind of rub it against the top of the container, and then we blot it on one of the biosquares to pull that all out of there. So then we're going to time it. Like I said, each of these has a different amount of time. Like this is a piece of paper, they're boards. We actually use the sides of the bottles. When we have people that don't do this as often, or we have like high school kids in, we give them the boards because it's easier, but you guys need to learn to navigate the bottles, so we use the bottles. At this use the seconds, most of our clocks, all of our clocks have second hands. That's plenty fine. It doesn't have to be, oh, I went 21 seconds. That's okay. 20 seconds is what we're looking at though. And then what you're gonna do is you're gonna hold it against between the strips, and so you're just gonna go back and forth until you find what it's closest to. When I'm grading these, I will grade you okay either way. Like if I say there was 6.5 and you say there was 7 and that's the next box over, you're okay. Because I know that there's always gonna be some variability. So whatever I read it as, I accept 1 on either side. So, so you just pick your best. I always have people come up to me, what do you think it is? Pick what you feel matches the best. Reporting out the results. Some results get reported out positive, negative. For example, leukocyte esterase. I'm sorry, no, not leukocyte esterase, nitrites. Positive or negative. It's one or the other. There's no in between. Sometimes you get a plus system, 1 plus 2 plus 3 plus 4 plus. We will never use the plus system in our class. I always want you guys, when we can, to use the semi-quantitative, use the number. Because that way we can compare, when we do our confirmatory methods, you can compare them with each other so that you understand what is a 30. Is that a 1 plus or a 3 plus? That doesn't make as much sense. A 30 matches a 30 or does not match 100. And then otherwise small, moderate, or large. That would be the other category that we would sometimes use. Every laboratory establishes how they do their own policies. You're going to hear this over and over and over from me. Whenever you go someplace, you follow their policies. Okay? You're never going to go someplace and say, well, that's not what Leah said. Leah said to do it this way. Every way is okay as long as you follow the policies of where you are at. Within this area, every hospital has slightly different reporting rules. So you follow the policies for where you go. And the last thing, when you do your clinicals that your preceptors ever want to hear is that's not how Leah taught us. If I hear you told them that, I'll be so angry. Because I keep telling you, every place has their own policies. So make sure you're following the policies of wherever you're at. Always follow manufacturer's directions. We talk about this in basic lab skills. I'm going to talk about it here. Anytime you get a new test in a lab, make sure that you look at the requirements and don't just trust. I'm going to tell you an example, story. At one point, this is many, many years ago, UW hospitals and clinics were getting all these negative rapid streps. And you know the strep, if it's negative on the rapid, they send it for culture. Well, then 2 days later, they get a positive culture, and then the doctors had to call in things, and it was delaying treatment by 2 days for streps. And what happened was my daughter came in with, we all knew it was strep. They swabbed her, and they came back, and they said, you know what, the rapid test was negative, but we're getting a lot of these false negatives. They turn out positive. This looks like strep. We're going to treat it. And I asked, what test kit are you using? What happened was these new test kits required a particular swab. And in the past, they could always just use the basic cotton swab. These test kits said specifically do not use a cotton swab. They hadn't read the new insert of the new test, and they were still using the same swabs that they've been using for 15 years. And when I brought it to their attention, they're like, oh, you know, it just got missed. So it's always important, if anything comes and changes, if they've updated anything, a new kit, you might have, you know, kit A, and now you're at kit B. Read through your procedures, read its limitations, read all of that. Same thing with urine strips. If you get an updated urine strip, a different brand, whatever, read through the inserts. Anytime this happens. It happened to me another time at UW Hospital when I used to work there. We used to use the plain red top tubes. Well, we went to the plastic. This is early on. So when I first started our career, everything was glass. Mm-hmm. That was also the time HIV appeared. Wink, wink, nudge, nudge, right? So everything, you know, they're like, oh, we can't do that, you know. So things started changing, so we got these plastic tubes in. Didn't think anything of it. It's still a red top. Well, what's on a red top tube these days? clot activator, right? So that clot activator is sprayed on silica, so glass particles. They found out that the red tops, the plastics weren't clotting because real glass has grooves for those platelets to kind of start congealing and starting, something to grab onto. The plastics were too smooth. So they sprayed them with this clot activator, silica, and I didn't realize it because I didn't read my inserts. And meanwhile, I'm working on a $1 million study that I hadn't validated the new tubes. Oops. That's the first time I learned. Anytime anything changes, read through your instructions. So you never know what it might be that might cause an issue. It didn't, thank goodness. It did not cause any issues. I was fine, but I had to revalidate those tubes to prove it hadn't caused an issue for the previous 8 months I've been using them. So read your inserts, read the directions, read the warnings, the limitations, handling, storage. Most of the time, things get stored on the counter. All of a sudden, this new kit might need to be stored in the fridge. You don't know. So it's really important to make sure you read all your manufacturer's directions. Storage. These bottles come with these desiccants. Some are little packs, some are in the lids. When you open them up, you're going to open it, take a strip out, put the lid right back on. You're not going to open it at the beginning of your shift and close it at the end of your shift. Every time you pull one out, you open it, pull one out, put the lid right back on. They get stored at room temperature. You got to keep it away from chlorine bleach. Thankfully, we don't use chlorine bleach to clean counters and stuff, but we will talk about why that's important later. Always note your expiration dates. The containers come with an expiration date on the bottom, but you also want to note open, the date, and your initials. That way, every time you open anything new in the lab, you write opened, the date, and your initials. Because everything you open has an out date from the time you opened it. So not only do we have the permanent expiration date, you also have an expiration date based on when you opened it. That being said, in our labs, everything is expired 90% of the time. It's okay for us. It's illegal in a clinical lab. That's why we get all this stuff. They give me all their expired stuff that we can use that saves us thousands and thousands a year. So it's okay. So you guys will note on a regular basis expired things. It's okay in our purposes. But in the real lab, it's really important to stay on top of that. They actually have to put it in a special area in a box that's closed up. This is like 4 Madison College, so nobody accidentally uses it. And then we always get calls right before big site visits and inspections. We got stuff in the fridge, come get it out, so it's not there when the inspectors get there. Thankfully, the inspectors understand that they are supporting educational facilities, but we just don't want to give them anything to raise an eyebrow at. Quality control. Per CLIA 88, we must run at least 2 control products, so a normal and an increased at least once every 24 hours. Follow the protocol where you're at. Some might require more. Some instruments might require more versus the strips. It depends on where you're at, what the requirements are, but per CLIA, 1 high, 1 normal every 24 hours. You must analyze them in the same method that you're going to analyze any other urine. It must just be run the same way. If anything is out of control, you have to solve the problem before you can run a patient. Starting next week, we'll actually be doing QC at the beginning of all the labs. If you guys find something out of control, like I said, we have expired strips, come see me before you repeat it. Because depending on what it is, I may decide the strips were too expired, or I might just say, nope, that's okay, and keep going.
It's not just for patient tubes either. If you're in a pharmaceutical setting and you're trying to figure out dosages and things, and you're working through a batch, it's in those settings too. They make you run the QC before you do the samples.
Good. I always wonder, when you get out of the, the lab setting, QC, it's it's this magic concept that may or may not happen. It's always kind of interesting. Okay, we talked physical, we talked chemical. The third part is going to be your microscopic. In our microscopic analysis, we spin it down, we look at the sediment on the bottom, and we're looking for cells, epithelial cells. Some epithelial cells are normal. Our body sheds epithelial cells inside and out all the time. This is why we do a midstream. A midstream is going to help flush any of those loose epithelials off so they're not in the urine. There are some epithelial cells, though, that are not good. And we'll talk about those when we get to this part of it, but there are some that are really important for us to catch because they can show early kidney disease and problems like that. We look at white cells, we look at red cells. So, the solid form cells we're going to look for. We're looking at casts. We'll talk more about what a cast is later, but we're going to look for those. Crystals. crystals are so much fun to look at, and thankfully most of them are pretty like okay and normal and don't cause problems, but there are some that are really big problem makers. So it's really important for us to be able to recognize and see those crystals. And then miscellaneous, we got bacteria. We're gonna see bacteria in our urines. We're gonna see parasites possibly, like trichomonas, yeast, candida, fungi, things like that. Sperm. We see sperm in urinalysis all the time also. So these are all formed elements that we're gonna be able to see under a microscope. So now we're going to talk about types of urine collection procedures. We're going to talk about midstream, pediatric, catheterization, and suprapubic aspirate. There are other kind of often in ones, but those are like the 4 main categories. There are like certain ones have subgroups under them, but we're just going to talk about the 4. Midstream clean catch technique. This is good for routine urinalysis and bacterial culture. This is why we have everybody do a clean catch, because if something does trigger a microscopic and a culture, we know it's a clean catch, and it's not going to be garbage in, garbage out. You must follow the CLSI standard collection guidelines. We got to cleanse the area. You're going to avoid the first part of the specimen into the toilet. Then you're going to stop, which always I think is funny, because if you've been holding it for a while, good luck stopping, right? And then you're going to collect it, and then you're going to pee the rest of the toilet. That initial flow is going to flush out any of the transient bacteria, those cells, anything that's just not really causing problems. It's just there. Because you got to remember, every time you have a hole in the body, things go into it, right? You know, our mouths are disgusting. We got our regular bacteria from our mouth, plus we got everything else going in. Every hole in the body is that way. Bacteria up through the urethra, it's just going to work its way up. And so we want to flush that out before we actually collect it, because we don't care about that little bit of transient. We want to know what's actually causing the problem. So here are our guidelines. Feel free to read those later. I always say a woman's clean catch requires 5 hands. If you read through it, it's hilarious. Hold this, do this with that hand, and it's like, okay, now do this. It's like, wait, both my hands are being used. What am I supposed to do? So it's kind of crazy, but you can read through that. Infants, we clean the area, and then we have this like little sticky pad thing that you put over the genitalia. Obviously, that's not going to be a clean catch. We're going to get lots of skin cells. Obviously, it's going to work better for boys than girls, right? Then you put this into the diaper, and you hope for the best. Yeah, I'm just going to leave that like that. It collects it. It's just not great. Catheterization. The catheter, it's a hollow tube. You pass it up through the urethra to the bladder. There's a couple different kinds of catheter. There's like a long-term cath, and then there's short-term like straight caths. Like for example, people that are paralyzed have to cath themselves on a regular basis to empty their bladder. They put it in, release the contents of the bladder, and then they pull it back out. Some patients are on a long-term cath where they put it in, they leave it in, and change it every so many days. Those ones have a balloon at the tip that they fill with saline, and it just sits at the bottom of the bladder, and so it just continually empties the bladder into a bag.
Going back to the infant stuff, I read in a fairly outdated book that you can like squeeze the diaper to get. Is that still a thing anymore? Okay.
Diapers have too many other things in them that are going to affect your results. They used to do that, especially if it's just like a regular cotton cloth diaper, maybe, but it's still going to have so many limitations, and it's going to get so many fibers in it, and it's going to be a lot of problems. So it's not ideal.
Okay.
But yeah, and think about all these new diapers. I mean,
The absorbency thing.
yeah, some of these things are freaky, like how much they hold. I was actually told you can actually use those diapers in, like, if you do like, I have these big barrels, and you can use the diapers in them because it holds water better.
Plants.
Yeah, so I did. I tore out some diapers and I put them in the bottom of this thing, and I'm like, okay, I don't know if it works or not, but I did it. Okay, so we talked about caths. The risk, this is a great way if you need to get a culture. You can just do a straight cath to get what's really happening in the bladder, but it's also a risk because it can introduce bacteria up into the bladder. So it's always a problem. One thing like if you get a trauma in a hospital, it used to be talking to you tube every hole, because you're going to put a cath in, you're going to put a tube in, you're going to, and anytime you do that though, you just raise the risk of infection because you put a foreign object into the body somewhere, right? So we try to be kind of careful. One of my claims to fame is I used to do a bunch of research on catheters. And I've been published on some of my research on catheters and trying to find ways. Because it used to be with this plain catheter, with a plain unimpregnated with anything catheter, a 10% risk per day that catheter was in of a catheter-associated urinary tract infection. So after 10 days, everybody had a urinary tract infection if they were in like an ICU. So we've done different things, like we have silver on them now. Silver prevents bacteria. Some of them are made with different products and things trying to prevent, you just put an open tube from the outside in, right? People who carry the bags with them, they put them up on the laps, right? Well, you just took all that old urine that's been sitting and put it right back up into the bladder. Problematic. So yeah, if you ever have a loved one that has a catheter, make sure to explain to them the importance of keeping it below the bladder. Because, they don't really do a great job explaining that. Or, to be fair, they may not listen all the time. It's hard for them to absorb, older people especially, to absorb all the information they're getting, but just help them out. Now, suprapubic aspirate, we actually put a needle right into the bladder. This is sterile. You do a sterile field, put the needle right in, pull it out. We use this on babies when we just can't get that clean catch and we can't figure out what's causing an infection. We can't get a good antimicrobial screen on them. We don't know what's happening. We'll sometimes do this. The other time, sometimes on pregnant women and sometimes on very heavy individuals that we just can't get a clean catch. Timings, timings, random is going to be our most common. You go to the doctor's office at 9 in the morning, 11 in the morning, 3 in the afternoon. Whatever time of day it is, you go to the doctor's office, that's when you're going to get your collection done. For most things, it's good. There's no restrictions. It's a good screening specimen, but it's not the best for certain things because it's not going to be the most concentrated. First morning is going to be the best when you're looking for a concentrated specimen. It's less convenient for the patient, but it's been sitting in the bladder. If you're lucky enough to go the whole night without peeing, for the whole night. You get to be in a certain age, you're getting up in the middle of the night to pee some more. But you get a longer stretch of time, hopefully without emptying the bladder. This is great for pregnancy testing. That's why the kids say do it first thing in the morning, because that's when those levels will be the highest, up until about week 3, 4, when it doesn't matter what time of day. But those early days where you're trying to find out early, first thing in the morning is best. Protein testing, nitrites, and bacterial culture are all going to be best for that first thing in the morning. Now, there's a couple different testing methods for urinary glucose. Postprandial, we're going to look about 2 hours after eating. By 2 hours after eating, everything should be pretty much back to normal. If not, we know we have problems. A fasting specimen, you void 4 hours after eating, you discard that one, and then the next specimen gets collected as a fasting specimen. Glucose tolerance testing, they do this on pregnant women. Thankfully, most pregnant women only need to do the one. If it's positive, then they go to the extended out. I think it's just a 1 hour usually. You drink this really concentrated bottle of sugar, and then you get your blood drawn and you give a urine before, you drink this off a bottle, and like 1 hour later you do both to see how your body handled it. If you pass that one, you're usually good to go. If you don't, then you have to go on one of the extended ones. 24-hour specimens, you talked about this in basic lab skills, right? Phlebotomy. One of the two. Excuse me. You first morning, you wake up in the morning, you pee in the toilet, you flush it away. You collect every urine all the way through until your first morning the next morning. You want to catch that first morning the next morning because that gives you the full 24-hour, including the concentrated overnight. This is best when what we're looking for varies based on the time of day. We use this a lot for proteins, creatinine clearance, and certain hormone studies we can use these for. Biggest problem with these, patient compliance. They have a big jug to keep it in the fridge. Of course, they don't want to keep it in the fridge, so they keep it next to the toilet where it stays warm, where things are going to be going on. We always suggest putting ice in a cooler and keeping it next to the toilet, even maybe. I ran out, I really had to go to the bathroom, so we missed one. I forgot to collect my next morning. It's really, really hard. The patient compliance can be the worst piece of that. That's why it's really important to tell them the instructions, emphasize the importance, and give them the handwritten instructions, or the written out instructions. These are what those urine containers, some of them look like. You gotta make sure they have the times on it, like what time they started, what time they ended, because we have to calculate the volume according to minutes. It's milligrams per minute. So minutes in a 24-hour day, it's 24 hours times 60 minutes, 1440. Sometimes it might be a 12-hour collection period. Again, you just calculate it. A lot of times instead of 24 hours, it might be 26 hours. It might be 23 and a half hours. You have to calculate it based on that. Basic math. Urine Container Requirements. These are actually spelled out what's required. It must be clean, inert, disposable plastic, should not be reused. Now, these are US requirements. Some countries have different rules. Must have a capacity of 50 to 100 mils with a round opening at least 2 inches in diameter. If transported, this is my favorite, must have a lid to prevent leakage. Anybody that's ever gotten a urine in the lab knows that we have not figured out how to do that yet. It's ridiculous, but yeah, they all leak. They must be sterile with lids. They must be labeled with a label that adheres during refrigeration, and it must be on the container, not the lid. Because lids can get mixed up. The urine is in the container, so the containers must always be what is labeled. Now, why this is a US thing, I taught in Tanzania for 3 months and was able to go to some of the labs there. They actually use glass vials, still, tiny little Erlenmeyers. People have to pee in the Erlenmeyer, and they log them in, they write their name, and they put a number next to it, and then they write that number with a China marker, so that's their identification system. Then they pour it out over the urine dipstick, read the dipsticks, and then they take that and they put it into a big bucket of bleach. So remember we talked about bleach earlier? Bleach is we're gonna talk about bleach on a lot of these strips because bleach affects results. If they don't understand the bleach affects results, which they did not until I talked to them, and then they started rinsing better. But they used to just do a quick rinse and then let it dry, thinking, oh, the bleach is okay to still be on there. It affects the results. So different countries, different policies. This is a US policy. In an unpreserved urine, the things we're going to see an increased results, bacteria, protein, nitrite, pH, and crystals. Those are all going to go up. Bacteria love urine. They're going to continue to grow. Protein goes up. Nitrites go up or possibly negative. We'll talk about why when we get to that lecture. But the bacteria take the nitrates to nitrites, and so it'll go up over time. pH will go up. The urine will become more and more basic upon sitting. Crystals start to form as the urine is sitting. Decreased glucose. The bacteria is going to eat that glucose as it's sitting, so the glucose will actually go down. Ketones evaporate. They're volatile. So because of that, we'll lose those. Bilirubin and urobilinogen. Remember phlebotomy? You collected that little amber tube because light's going to break it down. Same thing in urine, so it'll break it down. Red cells and casts are going to start disintegrating over time. So all those things will be going down in unpreserved urine. So preservation of urine, refrigeration works best. Up to 8 hours for sure. A lot of places might even go a few hours over, especially if you're like a clinic and the urine comes in at like 6 p.m. and the lab is gone. It may sit until 8 a.m. the next morning. There are chemical preservatives, and depending on the tests you're doing, affect which chemical preservatives you're going to use. Do not memorize this table. This is just to show you there's a lot of different chemical preservatives, and it depends on what you're looking at. This is why it's important for you to know what tests you're going to be doing, what you need to do. If you're going to be using, if you are doing a send-out, certain send-outs require certain testing setups, right? Remember from phlebotomy and basic lab skills, right? They have certain requirements. So you can't say, oh, it says use toluene, but I don't think toluene, somebody use thymol instead. They might be the same. They both start with T's, right? But they're not. So it's really important for you to know this when you're working. Do not memorize this table. You will not be tested on this table. You will never see this table again. It's just to show you that these all exist. And I'm sure there's way more than this. This is just some of the big ones. Key terms. Make sure when you're studying this unit, you go through all these terms. You know all these terms. Objectives. Make sure you go through all the objectives. All these objectives are also in the Urinalysis blackboard. You can print them out. It's a great place to study for your quizzes because they go over all of these things and all these questions. So make sure you know your objectives.